who reject things like fossil records, and literal creationists are the only exceptions. But in general, it doesn't say those things, and it's not a problem for people who are devout. In fact, Darwin himself was very religious. It's also not really something you can believe in or not. It's a theory. You either agree that there's support for it, or you don't. So moving on to that, let's talk about support for the theory of evolution, and where it comes from. We're going to talk about 4 major types of support. The first one is artificial selection, the second homology, both structural and molecular, the third fossil record, and the last physical observation. Artificial selection is probably the one that you're the most familiar with. Artificial selection differs from natural selection in 2 major ways. The first is that the selection pressure comes from the desires of another organism. Most of the time, that's going to be humans. So humans decide what traits they consider valuable, and they select for those traits. And the second is that these favored traits are usually not based on survival and reproduction, which is not the case for natural selection. An example of this would be how we select crops. For example, natural corn used to be extremely small, and even though it was edible, it wasn't particularly efficient to grow. However, humans have selected for different varieties of corn that are capable of growing in different environments, produce a lot more sugar, and are much larger. Other examples include things like the Brassica family, which produces cabbage, Brussels sprouts, kohlrabi, kale, broccoli, and cauliflower. Each of these vegetables was selected based off the desire of individuals to eat particular parts, be that terminal buds, lateral buds, stem, leaves, stems and flowers, or flowers themselves. You can also find traces of artificial selection in collected animals, like pigeons, for example, which are often bred to create fanciful varieties that would have a difficult time surviving in nature. Artificial selection shows that all of these traits are heritable, and if individuals are selected to breed with those traits become much more common in the population. So they support all of the underlying bases of natural selection. The next thing that we'll talk about is homology. Homology is the state of having the same or similar relation, relative position, or structure, and we call things that have homology homologs, and generally these things share a common ancestry. There are 2 major types of homology. One of those is structural homology, and the other is molecular. Structural homology has to do with the way things are structured, pretty much what it sounds like. For example, if we were to look at the forelimbs of a human, a cat, a whale, and a bat, we would see that the types of bones involved and the Hox genes that control the production of those bones are highly conserved. These are called homologous structures. They all have different functions, but they're located in the same locations in the animals, and they are made up of the same basic structures. We also see homology when we look back at the embryos that Anaximander was so infatuated with. It explains why early stages of development in different animals are very similar, even though those similarities might not be visible in organisms when they're adults. For example, both chick embryos and human embryos have pharyngeal pouches, which will eventually become gills in things like fish, and they have a post-anal tail. The tail will shorten and be reabsorbed in human embryos most of the time. These are considered vestigial structures, which means that they're remnants of features that were important in the organism's ancestor, but are no longer important for the organism's development. Sometimes these vestigial structures are not reabsorbed properly, and in the case of humans, the most common case of that is the growth of a post-anal tail. These are normally surgically removed before a kid is 5 years old to make it easier for them to sit and develop properly. Other examples of vestigial structures are the presence of both a femur and a pelvis in whales, which is an indication that their ancestors once walked on land, as well as limb structures like coccyx and pelvic girdles in snakes that suggests that their previous ancestors also had limbs. Usually the only visible remnant of this is going to be a spur that is visible right near the base of the tail. Sometimes students conflate the idea of analogous and homologous structures. So we've been talking about homologous structures. These are structures that have similar placement and similar structure to each other. Analogous traits have similar functions but have completely different structures, and they tend not to come from common ancestors. So examples of homologous structures are things like the forelimb in all of these animals: humans, horses, cats, porpoises, and bats. They all use the same combination of bones and are regulated by the same Hox genes. Analogous structures would be things like wings. The wings of a moth, a bat, a bird, and a pterodactyl are going to have very different structures. They use different parts of the body to create lift, and they do not have a common ancestor as a result. So remember, homologous structures are not the same thing as analogous structures. Homologous structures have similar structure and placement, and they share a common ancestor, whereas analogous structures have similar functions but different structures, and they do not come from a common ancestor. Not all homologies are obvious because they're not all physical structures. Some of them are molecular. For example, the fact that Hox genes are conserved across organisms, from a fly to you, means that they are homologous. They have similar structures and similar functions. Another example of this is how similar specific genes are. In this image, you can see the sequence for the mouse gene PAX6. PAX6 is important for eye development and eye control. The sequence similarity for all of these, from the fly, shark, squid, and flatworm, are pretty similar, but the proteins are identical. So not only does this gene perform the same function in controlling the development of the eye, it also produces the exact same protein. Let me give you another example. We'll talk about a disease that affects some individuals. It's called Parkinson's. Parkinson's is usually associated with a tremor, but it actually causes a lot of other symptoms. Things like problems with balance, increased stress and anxiety, thinking and memory problems, and sleep problems. This is caused by a specific gene called synuclein alpha, or SNCA. Mutations in this are autosomal dominant, and they cause Parkinson's, and sometimes they're implicated in Alzheimer's and Lewy body dementia. Here's an example of the sequence of this gene, SNCA, between humans, mice, cats, dolphins, and elephants. Notice that the vast majority of this sequence is identical. All of these organisms are capable of having Parkinson's, just like a human being. So even though you haven't seen an animal that has this disease, doesn't mean that it doesn't happen. Molecular homology and structural homology are a big basis of biomedical research. If animals weren't similar enough to human beings, we wouldn't be able to use them to learn information about ourselves. So you can thank this molecular homology and our relationships evolutionarily for all of the vaccines and other medicines that we've been able to test using animal friends and animal DNA sequences over the lifetime of genetics. Sometimes we don't have the luxury of being able to look at an animal that's alive right now or getting a DNA sequence. When that's the case, we can turn to fossils. Fossils are the imprints of remains of organisms that lived a long time ago, and they document differences between past and present organisms and can sometimes reveal the many species that have become extinct before now. In some cases, the fossil record also reveals historical sequence in which organisms have evolved. Part of what makes the fossil record so powerful is that it is essentially a timeline of events. As each generation happens, the next one occurs above it, and so the farther you dig backwards, the farther back in time you can go. Of course, to analyze them, biologists need help from geologists and paleontologists, so this isn't something that we tend to do alone. Many fossils can link early extinct species with species that are living today, and when new fossils are found, we can use those to rearrange how we think species are related. An example for that is how we know about the origin of whales. Beginning in the late 1970s, paleontologists unearthed transitional fossils that were thought to come from whale-related carnivores. These creatures look a lot like wolves, and they thought they were related to whales mostly because of their tooth structure and their skull structure. However, molecular biologists found close relationships between whales and hippopotamuses instead of wolves, and they hypothesized that whales and hippos were both descendants of the same cloven-hoofed ancestor. It's not possible to go back in time, so how can we know who's right? One thing that we can use is the structure of bones and how they are formed within the fossil record. For example, we know that there are two different types of hooved animals. There are perissodactyls, which have odd-numbered toes like the tapir, rhino, and horse, and there are even-toed hooves like cloven hoofs, which occur in pig, deer, camels, hippos, and pronghorns. These things have specific structures. For example, if we were to look at the knuckle bone, we can see that in odd-toed hooves, there are two humps at the top and we're missing the spur at the bottom. But in even-toed hooves, we can see that there's a double bump at the top, but there's also a connective spur at the bottom. Because whales and hippopotami are closely related molecularly, we would expect the ancestor of whales to have a cloven-toed ankle. So did the wolf-like organism have an ankle that looked like this? No. Instead, it has an ankle that's consistent with having an odd number of toes. So even though we don't have access to its genetics, we can say that it's not closely related to an hippopotamus, so it's probably not closely related to a whale. What about the other organism that might have been related? If we look at its ankle bone, Pachycetus, we can see that it's actually very similar to what we see in pigs and deer, which are cloven-toed organisms. This suggests that it's more closely related to the hippopotamus and more likely to be an ancestor to whales. This allows us to create a better timeline and include the correct organisms when we think about how whales evolved from land-dwelling animals to living in the sea. Do I expect you to remember all these details about whales? No, of course not. But what I'd like for you to get out of this information is that we can use the fossil record to try and give us information about organisms that we can't otherwise study. And we can use that to compare to organisms that are currently alive and see whether or not what the fossil record suggests relates to what we can see in the world today. In addition to that, we can observe things directly. There are lots of ways to document evolutionary change. For example, I already gave you information about how we can watch birds change in the Galapagos based on the amount of rain. But two common things that we talk about day to day is resistance to things like antibiotics and bacteria, and pesticide resistance in insect species. This is different from artificial selection. Artificial selection is selecting for a trait that has nothing to do with survival, but is something you want to happen. Under most circumstances, when you apply pesticides to, let's say, a crop field, maybe by crop dusting like in this plane, you're trying to kill everything. It's essentially a genocide on this insect. This is an extremely heavy evolutionary pressure. This means that only a few organisms are going to survive, those that are resistant to your pesticide. They are the only ones that are going to be able to have offspring, and that means that you're going to increase pesticide resistance every time you spray the field. This is the same thing that happens when people overuse antibiotics. If you don't kill everything and you leave some survivors behind, those resistant individuals are the only ones that can produce offspring, so the next round of antibiotics won't work. Remember, this is not an example of artificial selection. This is natural selection. Natural selection doesn't mean that humans aren't involved. After all, human beings are part of nature too. All of these examples of evolutionary adaptation reveal two important points about natural selection. The first is that natural selection is more of an editing process than a creative mechanism. This means that it doesn't just create spontaneous things. Instead, it makes small changes to what is already existent. The second is that natural selection is contingent on time and place. It favors heritable traits in a varying population that fit the current environment. That means that something might have been advantageous a few years ago that might not be advantageous today, but you're still going to see it. It's important to remember that natural selection does not create perfect organisms. It's only going to help produce organisms that are better at living in the current environment. Alright, now that we've talked about what evolution is and some support for it, let's talk about what a species is, so that we can think about how new species occur.